also, from the context of somebody sharing the data, one of the most rewarding to be able to give real-world data into the hands of our future practitioners to help them move forward. So has everybody gone to this link that intends to? Can I move forward? Okay, great. By the way, so that's a longhorn. He's not very long yet. That's an immature longhorn, but he's the mascot of the University of Texas. I think he's kind of cute. His predecessor recently deceased, but I think eventually his horns will be probably about 8 foot wide, which is pretty amazing. But they'll curl at the end so that. So my colleagues and I, a while ago, created the Neuro Bureau. And in the Neuro Bureau and the creation of that, we codified what we believed should be happening in the neuroscience community. And we believed that data, tools, and ideas should be openly shared. So we didn't know that that was open science until somebody later said, oh, I totally agree with your stances on open science. And Daniel Margulies and I, who we're the Neuro Bureau, we're kind of like, yeah, no, that's great. We're into open science, you know. I mean, when we started, it was kind of a... maybe like a social club for network researchers, kind of a network of network researchers. But that's sort of what began my foray into this field. And when you think a little bit hard about it, the goals of open science and what people tend to say about open science is that it will accelerate the progress of science. It'll make scientific results better through reproducibility and other things. And so I wanted to kind of give you a few things that I think of are key areas where open science can help in neuroscience, both clinical and cognitive. So, of course, it democratizes tools, access to tools, data, and education, right, that we need for our next generation of researchers. And I think that's one of the most important things we need to do is build up from the base that we have, build up and improve the quality of research that's done in this field through better education. It allows computational researchers access to data so that they can help solve our problems, right? So there's a story that's very common to those of you who have computer science background, but back in the early 80s, the United Postal Service, the U.S. Post Office, was trying to come up with a way to automatically route their letters. And the way that they came up with it is what they wanted to do is use computers to read addresses. But the technology wasn't very good at that time. So what they did in order to improve the technology is they released a database of handwritten digits from a lot of different letters, and they released the labels, what they should be. And now, or for several years after that, I don't know if it's still true today, that data set ended up being a benchmark data set that was used by everybody that developed prediction algorithms, right? So not only was it great because it spurred all of this research into a new area, but the UPS automatically knew how every new algorithm worked on their problem, right? So it's kind of a win-win. And hopefully we can build that same type of thing with neuroscience where we get our computational colleagues very interested in these problems. They use our benchmark data sets whenever they write a new paper, and we automatically know in the neuroimaging community how well those methods address our problems, which I think is great. Also, it's a great way to amass the resources we need to test our hypotheses, and it's more efficient use of funding resources and government resources, federal resources, tax dollars across the board, which is fantastic. So there are a lot of data repositories out there. Here are some of them. Chris and Oscar are involved with Open Neuro, which started off as Open fMRI. We'll talk a little bit more about that. But one of the questions that people might think of is, well, why do we need so many different repositories, right? So some of these repositories, so Open Neuro, 1000 Functional Connectomes, INDI, NDA, Connectome Coordinating Facility, are generic repositories where anybody can upload their data, right? Some of these others, Brain Genomics Superstruct, Ping, are specific data sets, right? Specific projects to generate data that just decided to share it on their own, to come up with their own repository. And if you think about it, this leads to kind of an explosion of the number of things out there, and it makes it a little bit more complicated for you, the consumer of this data, to be able to find things, right? But the reason I think that it's important that we still have a lot of different repositories is to help share the burden of sharing, right? So the amount of storage is very expensive, paying for the high-speed networking to allow people to efficiently get the data. All of these different things come at extraordinary cost. And the federal government, in of itself, is contributing some money to help out, but it's not taking up the whole slack, right? So what we need to do is amortize the amount of effort involved and the amount of expenses across a lot of different individuals. So every single one of these has their plus and minuses. They specialize in one type of data versus another type of data. But the thing is all of these efforts should be applauded. Within them, you will notice that some of them, they try to differentiate themselves, which is almost necessary in order to be able to continue their funding, right? You have to be able to explain to your funding why you should fund, my project compared to somebody else's project. But at the end of the day, it's, the goal is that all of these things are beneficial. All of them should be rewarded. So I kind of wanted to take you through, and I apologize that this is going to be from my perspective a little bit. I definitely include a lot of repositories that are otherwise, but these are the stories that I know best, and it's the easiest for me to tell. So maybe we could get other people's perspective as well. So the data sharing, the concept of data sharing in neuroimaging and repositories for it actually goes back quite a long way. So Mike Gazzaniga and Jack Van Horn started fMRI-DC at Dartmouth quite a while ago. And they had, at that time, Gazzaniga was an editor of a journal, and he had a mandate where if you shared the data, or if you published in that journal, you had to share the data through that repository. They were years, they were a decade before their time. There was a lot of backlash. Before its time. That was based on XNAT. It actually had a fairly nice infrastructure for being able to do it, but the community wasn't necessarily right for it. So the 1,000 functional connectomes, I call it sort of the beginning of sharing of neuroimaging data. And at this point, it kind of hit, I believe, at a time when people were starting to be willing to actually do this. And so it kind of, when it came on and the initial papers for that, they were sort of a special thing. So when this came out, this was 33 sites. It was over, even though it was a 1,000 functional connectomes, there was over 1,200 data sets that were involved in it. And this was started by Bharat Biswal and Michael Milham. And Bharat Biswal within the resting state community is seen as, a leader and one of the people that sort of developed the area. So a lot of people sort of kind of bought in for this. And their goal was to just collect a really large data set so they could look at things like sex differences and aging in these in resting state data. And a lot of people donated. And one reason why they donated is they got some big names in there, like Yufeng Zhang in Beijing and Randy Buckner, to give very large data sets. And then people were like, well, there's a lot of value here. We want to be a part of that. And they started sending it. So this was sort of, originally the idea was just to aggregate data from a lot of people so that we could build a bigger data set, right? Because no single individual at that time was able to collect 1,200 data sets, or at least to do it easily. And that's where it began. And then they decided to share the data. And from then, it just kind of skyrocketed. We'll talk more about some of the publications related to this later. But so one of the issues that was quickly apparent with this data is albeit very useful, is that it's a bit limited in what you could do with it. All we knew about the individuals was their sex, their handedness, and their age. And we really didn't know much even about the scanning parameters that was used to collect the data. I tried a project where I wanted to get all of those parameters together, and it failed because, for whatever reason, maybe organizationally, the people didn't really have that data. So Mike kind of went back to the drawing board and he said, well, what we need is a repository where we know more from the data, where there's more interesting problems can be solved. And so from there came the International Neuroimaging Data Sharing Initiative. And this has been very successful. The goal of this is to have data with phenotype, right? So there's more than just sex, age, and handedness. There's at least one other measure, be it a clinical diagnosis, be it an IQ score, something of that nature. We mandated that all the data has to have all the parameters so that whomever's using the data can have access to all of that information. And so this has been ongoing. So in this, we have both there's both prospective and retrospective data sharing. Prospective data sharing means that the data is being shared as it's collected, and in many cases pre-publication. The retrospective data sets tend to be kind of the data sets that have been sort of sitting around, have been used before, and people are kind of sharing them after that. So right now there's about 35 different projects within here. Some of these projects actually have data from many different sites, so it's a bit harder to calculate all the sites that are involved. There's about 10,000, at least 10,000 participants. We now have a non-human data set I'll talk more about. And now, although it began with connectomes data, it began with resting state fMRI, it's much more broad now. We have quite a bit of different data types. And one of the things that is very important with Indy and may differentiate it a little bit from other things is this idea that the data has to have some sort of assessment along with it, right? Some additional variable that we're interested in. So as a part of Indy, Mike and others began this consortium model. So the consortium model is very similar to what the 1,000 functional connectomes is, but it's where a bunch of like-minded researchers compile all of their data together, and each one of those researchers gets the benefit of everybody else's data, but it also builds a data set around a specific theme, right? So again, this is kind of motivating individuals to sort of access this data, use the problems that people are interested in. So this started with the ADHD 200. There are 8 sites that put in data. There's 386 ADHD, 535 typical. And this is primarily resting state fMRI and structural MRI. That was followed on by the ABIDE. There's been 2 releases of ABIDE, ABIDE 1 and ABIDE 2. And overall, it's up to over 1,088, sorry, that's a typo, autism and 1,200 typical. So these, when they began, the ADHD 200 began with a competition that encouraged people to try to come up with the best classifier of ADHD, and that spawned a lot of research into using classifiers with neuroimaging data, and the ABIDE is kind of to brought that on. So these, again, as I said, these data sets are kind of a distributed model, right? So everybody is giving data that they've already collected. So it is completely, it's different. Right? It wasn't organized from the beginning. The scanning parameters vary quite a bit across site. There's a lot of technical variation that needs to be dealt with. And we'll hear a lot more about how to deal with that in the later talks today. But it begins to ask the question, well, so these things, they kind of highlight some of the issues that we have with our methods, but they're not necessarily optimized for learning more about methods development, right? And so this was followed on by the Consortium for Reproducibility and Reliability. And these are a variety of different test, retest data sets. And the goal is that people can use them to look at reliability, reproducibility, those measures, and be able to optimize their methods around those ideas. So there's over 1,629 healthy individuals involved in this. Again, we don't have a lot of information about these individuals. It's more age and sex again, but we have more than one scan on each one of those. So overall, there's about over 3,000 MRI scans and over 5,000 resting state scans, over 1,500 diffusion scans that are associated with this data set. There's a variety of different protocols or different experiments that were used in here. There was one participant that was scanned 100 times. And there are 2 participants that were scanned 5 times a day for 3 days. There's one guy that was scanned every hour for 24 hours. These are some very large data sets, and there's some interesting stuff in here. So it'll allow you to test a lot of hypotheses about maybe diurnal variation or scanner test retest stuff. So that's available. We kind of moved on. The use of open data in human neuroimaging has really helped advance a lot of the methods. And methodologically, in non-human primate and rodent research, methods have languished a bit. And there are a lot of hard problems, right? So solving problems for human data doesn't necessarily solve the problem for non-human data. So they just need to be reevaluated. So recently, there's been a consortium called PRIME-D, which is a primate data exchange, where we have 25 international sites that have contributed data from 98 animals. And so structure, functional, DTI as well. But now we have a variety of paradigms. So there's anesthetized monkeys. There's awake monkeys. Has anybody here scanned an awake monkey? It's very difficult. The people who train monkeys to be able to tolerate the scan, they're like, I guess monkey whisperers, they're amazing. We also have a lot of movie watching data on these monkeys. And there's movie watching data where they're watching movies that are the same movies that we have human watching data on, so we can look at neural correlates across species, which I think is kind of cool. So OpenFMRI is something that hopefully you've all heard about. So this came on maybe just a little bit after the 1,000 functional connectomes, and it was, whereas the FCP was initially focused much more on resting state, this was initially focused much more on task-based data. This is started by Russ Poldrack, the lab where Chris works. It's now been Open Neuro, which hopefully y'all know more about. So currently there's 122 distinct data sets, over 4,000 individuals, 208 different tasks, but it's much more than task fMRI as well. There's resting state data, there's PET, EEG, MEG, DWI, and they have actually, Open Neuro has a beautiful interface. Y'all have done very well in terms of being able to access the data and things. I think it's nice, but definitely something that's very interesting. So a data set from there that I'd like to highlight is it okay to say who the individual is? Is that well known, or because on the page it doesn't? Okay, so Russ Poldrack, the PI on the project, scanned himself 107 times, so 3 times a week for I guess several weeks in order to be able to do it. So this includes T1, T2, DTI, resting state, and a couple of different tasks. Pre-processed variants of this data is available. Pre-processed data. So with Open Neuro, I'll talk a little bit more about that, but a lot of the data is available pre-processed. There's blood samples in there. I don't know if the blood sampling data has been processed and released yet, and several different experiments that he did. So this is a very interesting project that I encourage y'all to look at. I think it's a really cool one. Another one, so I have a bit of a clinical bent to neuroimaging. So another one of the projects that I thought was really exceptional is this Consortium for Neuropsychiatric Phenomics. So this is out of UCLA, and they're trying to get sort of a broad understanding of neuropsychiatric disorders. And so here's this idea of phenomics where you're sort of studying these individuals from the gene to the syndrome level all the way up. And so there's quite a bit of information on here. The individuals are well characterized in terms of their phenotype. So they go through psychiatric screening, diagnostic questionnaires, all of those things to verify that they have a diagnosis or no diagnosis. So there's 130 controls, 50 schizophrenia, 49 bipolar, 43 ADHD. I imagine that this sample is probably still growing. Is that right? Okay, it's completed. But so as I mentioned before, this is on Open Neuro, so you can access it either way. So that's a really cool one. But another one that Chris pointed out that I think is kind of really cool, highlights the benefit of both of these data sharing repositories, is data from very important papers that have been released, right? So this is a Jim Haxby's paper. So this pretty much defined, the methods used on this paper pretty much defined this idea of MVPA and using these types of pattern-based approaches to analyze neuroimaging data. It also started a huge debate over the sort of the neuroscience and the cognitive processes behind face detection, right? So it's amazing to be able to have access to this original data so you could directly test the replicability of those results using different methods, using the same methods, or test the variation on the hypotheses, right? So you can go to the data and see if it's there. So this, I think, is a really cool example. A really important aspect of data sharing is getting the old data out and available so that we can all reevaluate some of the claims or see how our own special methodology or hypotheses apply to that data. So that was a really good suggestion. So most of the things I talked about before are data sets that weren't necessarily created with the intent to share, but they ended up being shared in sort of this ad hoc aggregated way, right? But on the other end of the spectrum, we have centralized prospective data sharing. So maybe my connectome and the other one actually apply to this as well. But and so there's a couple of projects like that. And the nice thing about this is there's much less technical variation in the data, right? So the TRs are the same, the parameters are the same, the scanners may be the same or very similar to one another that are used in it. So they're very nice, maybe from the neuroscience perspective, but if you want to study how to deal with heterogeneity in the data, maybe they're not as nice. But there's some really cool projects here, and I'd just like to highlight a few. So the Rockland sample is a project that I've been involved with in New York, and this is a combination of 4 different ROIs that have synchronized their data collection efforts, both their assessment and their neuroimaging data, to build just a really large repository of data. So there's individuals in here from 8 to 85 years old, and rather than having a specific exclusion or inclusion criteria, we just kind of took all comers. comers, right? So if an individual came in that they could be scanned and they weren't going to kill us, then we would involve them in the study. And so when you have that level of sort of variability, you need to be able to characterize it. So we do very deep assessment on the individuals so we know, right? So we may bring in somebody with depression and 5 other comorbidities, but we assess that and write it down so it's available to you. So we kind of think of this as more sort of studying individuals in the wild, right? A lot of times when we do, clinical neuroimaging studies, we'll take unicorns. We'll take very clean, healthy control individuals and compare them to very clean, maybe depressed individuals who have no comorbidities, right? And whereas that is very smart if you want to maximize the power, hopefully the power, to detect differences, it also may not characterize how people are just every day, right? So if you have one psychiatric disorder, odds are you have more than one psychiatric disorder, right? There's a lot of comorbidities. There are a lot of things that are known to be linked, like anxiety and depression, for example. So hopefully that allows us to get at that. But also one of the goals was just to get as much data as we could to create a sandbox for our colleagues to play in. So we have IQ measures, we have physical measures, we have blood draws. We haven't processed the DNA on that yet. Cognitive tasks, psychiatric assessments, just the whole kit and caboodle we kind of threw in on this one. There's not necessarily everything though, so we didn't evaluate their smelling ability, but anyway, hopefully this will be a good place for you to start. So if you are interested in a population that is relatively common in the wild, then there's likely to be enough of those in this data set for you to get a nice chunk of individuals to look at, right? So in terms of the characterization of the individuals, the most common diagnosis is probably some form of substance, either use or abuse. And secondary to that is depression. In our youth, the most common things are ADHD. So it fits very well with what we'd expect. So in this data set, we have it started off with 1,000 individuals, kind of from 8 to 85. That was a cross-sectional study. That's mostly been completed. Now we have a child longitudinal where we have 180 young individuals that are being scanned 3 different times. I think it's every 18 months, to look at developmental trajectories. We also have an adult longitudinal, which is 40 to 85. Again, they're scanned 3 times. And that includes a gold standard measure of cardiovascular fitness. And that's to look at the effects of cardiovascular fitness on cognitive aging, but also you can use it to map aging trajectories, which is nice. I have my study in there is 200 participants, and that's a neurofeedback study looking at modulating the default network. So that data is in there as well. So at the time, we kind of used the best data collection that we could. So this came out around when the Human Connectome Project was founded. It has the same program officers, so they ask us to use the same protocols that were being developed for the Human Connectome Project. So it has multiband imaging, it has a nice DTI. It's meant to be a bit of a broader sampling in terms of the imaging. So that was a very nice project. And then Mike Milham, who kind of led this, and some of our colleagues, we decided to create another project, right? And so when you think about neuropsychiatric disorders, there's a lot of heterogeneity in the data. And if you think about what we want to be able to do with neuroimaging when it applies to these, we would like to be able to diagnose, but being able to diagnose, for example, depression, isn't necessarily something that we need a machine to be able to do, right? Doctors seem to be doing pretty well with that. But areas that are more important are looking at the difference from maybe somebody with autism and ADHD, right? Are these differential diagnoses when somebody's kind of on the line and we want to try to find out more nuanced information to be able to differentiate those. If you look at most of the classifier studies out there, we really can't measure their specificity because we're always just looking at healthy control versus the disorder. We're never looking at healthy control disorder 1, disorder 2, disorder 3. And the reason we don't is we don't have the data to be able to do it. And the goal here is to be able to collect that data. So these individuals are specifically recruited. So our goal is to get 10,000 young people aged 5 to 21 from across the New York metropolitan area. Again, it's cross-sectional. We do like a 4-day assessment on these individuals. We have so again, it's too small to read. I encourage you to look at it. And we have a very sort of a multi-modal approach to the imaging. So we have a resting state. We started introducing movies just because they're a lot more tolerable for children, and they enable a much broader range of analyses that can be done. So to start that off, we did an evaluation in 13 individuals of the retest reliability of watching these movies, which is another data set that's available as a part of this. But we also have things like an abdominal scan, so we can look at visceral fat. We have some quantitative MRI, so we could look at, maybe a myelination or things of that nature. So this is kind of a interesting data set from that perspective. Right now, I think it's on the order of about 1,500 individuals. It'll grow to 10,000 over time, but that first data set's available. So as I mentioned before, these are children, and it's hard to motivate people to bring their children in to get scanned, and we specifically want to have a lot of disorders. So this data set is what we call enriched for these disorders. So when we advertise for this, we say literally, are you worried that your child is suffering from a mental disorder? Come and we'll help assess that, right? So all of these data sets, the healthy controls are probably just sub-threshold, right? There's still some indications that they may have some disorder. So if you're looking for a clean, healthy population, that's going to be much fewer of the individuals in this data set, but there's going to be a lot of data sets where they have disorders. In this one, we also have EEG. So again, I'm sorry, it ends up pretty small, but we did quite a bit of assessments, including things like sequence learning, eye tracking, resting state, inhibition, naturalistic viewing, a couple of different things with the EEG that's also available in this data. So in these, you have structure, function, and function fMRI, function EEG. So hopefully that will kind of help more broadly. So everybody's sort of familiar with the Human Connectome Project, or I assume y'all are all familiar with the Human Connectome Project. It's gotten a lot of press. Well, after the first Human Connectome Project was done in 1,200 individuals, many of which were twins, but they're all probably about, like 22 to 27 years old. They're a fairly tight age range, fairly small variability too when it comes to a lot of demographic factors we may be interested in. Socioeconomic class, IQ, things of that nature, education. But that is so that was sort of the pilot for a lot of these advanced methods, right? So advanced DTI methods, advanced fMRI methods that were developed. But since then, it has been expanded to include a lifespan. So there's 4 cohorts from different age ranges that are available now through that project. There's also a variety of disease populations: Alzheimer's, dementia, epilepsy, anxiety, depression, psychosis. So there's it's become much broader. So the Connectome Coordinating Facility, it's humanconnectome.org, is very interesting to this. One that I didn't include in the slides is a UK Biobank, which some of our colleagues here work with, which is a much broader. I think it was 10,000 overall is the goal, 100,000 for the MRI assessment. And then it's part of a larger thing where they have other assessments on, over 1 million? Half a million. So it's huge, right? And so that's a very interesting project. You probably see Carla Miller had a very good paper on that. So it's definitely something I'll encourage you about. And I believe it's talked a little bit more about later today as well. So sharing raw data was sort of the first step, right? And that was a very important step, something that had to happen. And it helped out a lot of people. But, if you really think about it, if our ultimate goal is to really engage people who are not neuroimaging experts into the analysis of fMRI data to help us learn more about how to do these things, or MRI in general, or EEG data, neuroscience data, then we need to sort of reduce those barriers. And the biggest barrier that exists, particularly for neuroimaging data, is the pre-processing that's applied, right? Anybody here, many of you probably participate in methods research for pre-processing. It's kind of a mess. There's no clear indications of what's the best way to go. And if you are a machine learning guy that wants to look at classifiers on this, you don't want to have to wade through all of that and spend essentially enough time getting an additional PhD in neuroimaging in order to be able to do your research. So there's a lot of pre-processed data that are released for that reason. So a project that I was involved with is the pre-processed Connectome project. But now OpenNeuro has a lot of pre-processed data. The Brain Genomics Superstruct data has FreeSurfer that was generated on their data. Ping has FreeSurfer. The Connectome Coordinating Facility has data processed at various levels that you can access. So this has become one of the areas. It's a smaller component of the amount of data that's shared, but it's data that's readily available. So again, I mentioned with the ADHD 200, they had a classifier competition to do it. And so some colleagues and I in the NeuroBureau pre-processed all of the data and released all of those derivatives so that the teams that wanted to participate didn't need to worry about that aspect of it. And the nice thing is that the winning team used the pre-processed data, but also this data has been used quite a bit by a variety of people. We followed that on with the Abide pre-processing to make that data more accessible. But what we did with this is we wanted to make it it much more rich for people to evaluate different processing decisions that they may make. So we process the data using 5 different functional pipelines, and each one of those pipelines, or I guess 4 different functional pipelines, and 2 or 3 of them are structural, so both, right? A lot of function and structural pipelines. And we have some variation in the types of parameters that are used, some of the steps that are used, so people can begin to ask questions like, how would my answer change if I did global signal regression, or if I filtered the data, right? So these are important things, but would be substantially difficult to do if you had to pre-process all, 1,000 data sets or so. So OpenNeuro is what OpenFMRI has become, and this has a lot of pre-processed data, but it also is a computational platform that you are free to use as long as you're using data that's shared there, or if you're using your own data, as long as you're willing to share your data on their timeline, which is a very liberal timeline. I think it's 3 years now. So I think this is, again, as I was mentioning before, it's a beautiful interface for it, and it allows you to execute a variety of different pipelines on a variety of different neuroimaging data. And I'll talk a little bit more about some of that technology. So anyway, most of the data that's available in OpenNeuro, I believe, is available at least one preprocessed form. That's probably fair, right? So another thing, so we talked about, so the OpenNeuro data, the data in the PCP, those are mostly what I would consider automated pipelines, right? So you pre-process all the data, you find the data that is good, and then you use that data, right? And the data that's not good, maybe you don't spend a lot of time really working with it. And if you think about what you want to do, if you want to be able to fix these things that don't, that are automated, right? You want to improve automated skull stripping or lesion segmentation. One thing that you need is those expertly applied labels, right? So there's some data sets that have been leaved, and I realize this is very myopic. There's a lot of data sets that are out there from various competitions and things. But another concept is sharing data that has already been sort of manually assessed and labeled. And this is great for any sort of classifier research that you might want to do. So an example of this is Atlas, which are 229 T1 MRI scans. So most of these are collected in the clinics, right? So this is medical data, which is a lot dirtier than a lot of the data that we do with our research. And if you look at this, you'll see why. But things like they may be, you'll have 1 millimeter in plane resolution and then 5 millimeter thick slices, for example. So things that may not completely work with our quantitative tools, but this is our target, right? If we want to apply things clinically, these are some of the data types we have to deal with. But here, these are manually traced lesions in this data that you could use as sort of training data for whatever you're interested in. Or you could directly test your hypotheses about stroke with it as well and not have to do the manual editing. Through my lab, we also released skull strip data where we manually skull stripped, and you can use that to help improve your classifier skull stripping techniques. So this is another kind of thing that I think is important to look for out there. In addition to these, I would look for things like Mackay and other data sets. There's some manually editing in there. And so, we're kind of building up. So we had raw data, we had automated preprocessed data, we have manually edited data, and then there's repositories that include the next level, right? So after the data has been published, for example, so neurosynth.org is a way to do meta-analyses, and the approach, so this is Talia Coney, who's a colleague of mine at University of Texas, but the approach is that they they read papers and try to infer from the paper using a computer, right? So not human read, but computer read, where the locations of the activations are, as well as what contrast those activations go through. And then you can do very large meta-analyses. So this is a meta-analysis of pain, I believe. Yeah, 420 studies of pain. So currently, they have in their database over 413,000 activations reported from 11,000 different studies, right? So there's another database out there that's called BrainMap, which is a very nice database, but it's hand curated, right? So people hand read the papers, and then they transfer that information in there. And people have done comparisons between the meta-analyses you do between Neurosynth and those hand curated, and you see that there's actually a very nice convergence between those findings. So this is kind of the quick but dirty method, where you're relying on very large numbers in order to kind of handle that noise. And the BrainMap is the other approach, where it's very clean data that you put in with the hope that it improves it. But so this is a very nice repository that I encourage you to look at just to start developing your hypotheses or testing your hypotheses against what has ever already been published in the literature, right? And so then you can think about, well, rather than reading it from the paper, why don't you just read it directly from the statistical maps that people use to develop it? So Chris developed a repository that's called NeuroVault, where you can put in your unthresholded statistical maps when you're done with your analysis, and then be able to do many of the same things that you can do with Neurosynth. And actually, Neurosynth and this are directly connected to one another, so you can see sort of how your findings relate to other findings in the field. So that's kind of some of the nice things that you get out of this. It does very beautiful visualizations, so people will upload their data and use it to generate their figures, which is very nice. But also it allows you to do reverse inference and other things using the NeuroVault, using the data that is currently in papers and in Neurosynth to learn more about what your activations mean, right? Which I think is a really kind of compelling idea. And there's also direct support in, is it SPM? There's direct support from, or is there direct support to just put, I know your beautiful little button thing from a long time ago. Yeah, so SPM has a way to directly import your results into NeuroVault as well. But it's a nice interface. I think you could do this fairly easily. And so if you want to learn more about these things, we talked about the beauty of having all of these repositories, but the danger is it's just hard to find them. So Nitric is an amazing index of tools and data. So it's essentially, it's been around for a very long time, and it's kind of the go-to clearinghouse if you're looking for a tool to do something, right? So if I want to get into another area, say spleen imaging, and I want to learn about how doing spleen co-registration, I can come here and look for tools that have been presented there. So it's a fantastic way to find these things. They have a data repository themselves, the Neuroimaging Data Repository, and they've mirrored a lot of data from other sites, which is another very important aspect, so that we don't have a single point of failure to getting access to this data. They have ways of helping you with your cloud computing, but also I think their role as an index, the yellow pages for these various things, is really nice. And if you have a tool that is not indexed on here, I encourage you greatly to do that. That's very useful. So, we've kind of gone through where to look for tools and all these other things. A great way to kind of look for information and to learn and to meet colleagues who are like-minded using data or otherwise is to come to brain hack events. So the OHBM hackathon, we just got back from it, and we're all half the speakers were there. That's a bit of contributed to our fatigue. But this is a way to interact with other people who are interested in open sharing and to learn from them how to use their tools, and also to help them develop their tools or get them to help develop your tools. Okay? So this is another area where I'd like to take a brief poll. I would have used some fancy technology if it was available to me. So y'all have heard about all these things. Hopefully y'all are excited about them. What are some things that y'all think are limitations to you using this data, or limitations that limit the sort of the impact that you can get with this data? Does anybody have some concerns about using this data that they'd like to share? No?
Don't be shy.
Yeah. So the ones that I've heard most about are, well, you can't use, so they'll say, well, this data is not that exciting, so you can't get a high impact paper with it. Do y'all think that that's likely? The impact of your paper? Well, that's great. You don't have to worry about the rest of my slides. Some people are worried about how hard the data is to use, right? That if the data is there, you don't know about how it's collected, it may be kind of a pain, right? So these are some of the things that are constantly used. So an effort led by Chris, again, the brain imaging data sharing structure is here to at least to try to fix the problem of data organization and trying to create kind of a common language that we can share data in so that you could easily and quickly pull it in, understand it, and effectively use it in your analyses. It includes both the folder structure and a naming structure that makes the data machine readable, makes the information from the data machine readable, but at the same time preserves the human readability of the data. And the nice thing, and so not only is it a way to share the data, but also it's a great way to organize the data for your personal data, regardless of whether you want to share it, so that when you have like a postdoc transition or you haven't analyzed the data in 5 years and want to come back and look at it, you know how it was collected and all that information is there, right? So one of the things you see, it uses the nifty format, which is kind of a standard in the field, a de facto standard, as well as an intentional standard. One of the drawbacks drawbacks about the NIFTY standard is you lose a lot of information from the header about the parameters used to acquire the data. So they replace that using JSON files, which is another human-readable and machine-readable format that includes all of those parameters. So the nice thing is once you preserve all of this information, you could build software that looks at that data and completely configures itself based on the information it pulls in, right? So when you're doing DTI, you don't have to worry about reformatting your vector files. When you're doing, distortion correction, you don't have to worry about calculating your right echo spacing, all of these things, right, which can be challenging, particularly when you're working for data without that comes from somebody else, right? So Chris and his colleagues have created another idea around this called a BIDS app. So a BIDS app is software that's specifically designed to understand the BIDS format. There's several different pipelines that are created. These are containers which help deal with the distribution and the installation of these. So if you wanted to run and compare, say, for example, CPAC and fMRI prep, you could quickly do that using the same data without having to reformat the data or tell the software anything, right? You tell it your processing parameters, but other than that, any information that it needs to know specifically about the data, it can get from the data, right? So I was a bit prepared in case y'all were thinking about the impact of data, right? So a lot of people that I talk about, they'll say, well, the nice thing about data is sharing is it makes data available, but a problem with it is you'll never get a PNAS paper with that, right? Well, in fact, there's been several PNAS papers published with it, right? So I wanted to talk a little bit about the impact that data sharing has already made. And this is through the perspective of the ND data, but there's other, I don't think that this is exclusively about ND data. I think that this information will generalize. So since the 1000 Functional Connectome project, if you look at all the papers that are kind of under this umbrella, all the data sets that are on it, it's generated over 913 publications. And that gives you kind of the growth and the breakdown for the different ones. 1000 Functional Connectomes has by far been the most published, but it's also one that's been around quite a long time. So if you look at publications by field, you'll see that the majority of the publications do tend to be in neuroscience-centric fields like general neuroscience, neurology, psychiatry, and medicine, right? But beyond those three, everything here down are computer science, mathematics, and those other ways. So there's really getting a broad reach of those things. So not only is it still valuable to the people who are in the community that could generate their own data, putatively, but it's also been valuable in other areas where they don't necessarily have that data. And you can see our breakdown of the publications by types. The vast majority are peer-reviewed journals, followed by preprints, which we presume will eventually become peer-reviewed journals. But we're really proud of the number of theses that have come out of this. There's at least 58 that have been out there. That number is a little bit harder to track. So then if you look at what is the impact of these things, right? So there's been at least 12 publications in PNAS, which is a very high impact journal. One of the most sort of popular papers or unpopular papers of the last 2 years is the cluster failure paper by Anders Eklund, and he used the 1000 functional connectomes data for that. So there's a lot of value in that data, even though that wasn't that phenotype, and it pointed out some failures in the way that we were doing things. So a really kind of impactful thing that has been accomplished with this data. Some of the questions that we face when we're under review is well, how much do contributors actually get back from this? Do they just kind of put their data out there and it doesn't really benefit them necessarily? But what we find is then, in a large case, contributors are actually using their data, combining it with other data from the consortium, and actually getting a much larger data set in order to do their analysis on, right? So it's helping them as well. So it's useful both to the sharer and the non-sharers alike. So then there's some questions that people say, well, okay, I'll concede now that that this shared data can have impact, right? So it has had some PNAS papers, but if you had just done any ADH, if you had used data you collected yourself, you probably would have a higher likelihood of having that, right? So we try to do a comparison where we look at sort of a CDF, cumulative distribution function, where we look at the citation score of these papers and look at the fraction of it. And what we see is that there's there's a lot of similarity between the distributions of citations for papers on autism that are from the Abide data set and ones that are from the non-Abide data set. If anything, I think you do a bit better with the non-shared data. And I think that has to do with the size of the data sets or the fact that you're doing a replication study, which may be perceived as having a little bit higher public thing. And to avoid just sort of an overly myopic sort of indie perspective, we also looked at Human Connectome Project and saw that we kind of get a lot of similar results. It may be a little bit of a bias when you look at the ACP investigators. A lot of the really initial ACP papers had a lot of impact, and that may be driving some of that. So we got 12 seconds left.